Hello and welcome to our Lakeside chat. We are David Wofford and Katie Noonan, co-chairs of the Rotary Nature Center Friends. And we're so happy that you can join us tonight.
As we begin our program, it is important for us to acknowledge that we are on stolen land, that Lake Merritt is part of Ohlone territory. We hear now from Corinna Gould, spokesperson for the Confederated Villages of LaShawn and co-founder of Segurate Land Trust.
Good afternoon, relatives. My name is Corinna Gould. I am the spokesperson for the Confederated Villages of LaShawn. We are here today at what most folks think of as Lake Merritt. We are in the territory of Huichin. Huichin is actually a territory that encompasses 6 Bay Area cities: Oakland, Berkeley, Alameda, Emeryville, Albany, and Piedmont. And so this is a place that my ancestors have been since the beginning of time. And this was a place of abundance. I'm so happy that people from all walks of life that now come into our territory can enjoy this beautiful place that my ancestors have enjoyed since the beginning of time. My relationship to the land that I have been born to, raising my children and grandchildren here, has been to tell the story, the truth of what happened on this land before other people came here. I'm hoping that as we begin to learn these lessons of fires in California, the pandemic that's happening, that human beings come back to living in reciprocity with the earth.
Our first guest tonight is Dr. Brian O'Lee, a fisheries biologist, and I'm so glad he can join us tonight because we've had some interesting events in Lake Merritt. We've had Chinook salmon appearing in the lake over the last couple of months. And so, Dr. Lee, can you help us understand what's going on here? This is something we haven't seen much before.
Yeah, I think it's pretty exciting, Katie, and thank you very much for the opportunity to talk to you. I think what we're really seeing here is a phenomenon that I think is always exciting, and that is this is a colonizing activity on Pacific salmon. The salmon in question is Chinook salmon, Oncorhynchus chowicha, beautiful name. And it's really an example of evolution in action. And it's just the first part of that in terms of what you're seeing in Lake Merritt. They're attracted to velocity and flow at the outflow of Lake Merritt through the tide gates. It stimulates them. They know they need to move upstream to spawn in spawning gravel. And they're anadromous fish. That is, they come back to freshwater to spawn and spend a great deal of time in the ocean, maturing and growing to a very large size. And Chinook salmon are really also called king salmon. So they're the largest of all the species of salmon. And they have the capability of digging redds in the gravel or nests and burying the eggs and fertilizing them externally and then burying them in the gravel. They stay in the gravel for a couple of months and come out and feed in the stream for a while, maybe 2 months at the most, and then leave for the ocean. So they're supremely adapted to some of these California urban streams that are dry in the summer because of lack of flow, and have cooler temperatures in the fall. So they're fall Chinook salmon. They return in November, December, to a certain extent, January. And so they're kind of a wonderful fish to identify. So you've seen the first phase, which is the migration into freshwater. So as I understand it, and I saw some pictures from Katie, which indicate that they were in Lake Merritt and they died, and there was a carcass there and I couldn't see an adipose fin, the little vestigial fin in front of the tail. So they're a hatchery fish. They were marked in a hatchery, but there could be wild fish as well. So it's a very exciting opportunity. It's the kind of thing we're seeing in Los Gatos Creek down by San Jose, and with Chinook, larger numbers. The unique thing about salmon, some of the salmon anyway, Chinook salmon as an example, they die after spawning. It's a form of parental care, and their carcass then breaks down and is utilized by fish and many mammals and birds. So it's an exciting thing. The trees take up this marine nitrogen as well, and they can trace it in trees adjacent to the watershed. You don't have all of those systems, that part of the life history yet. It's a possibility. Many of these urban streams have been cleaned up, and spawning gravel and rearing areas have been made available or created little opportunities from an engineering point of view. And so the fish have a hospitable place to spend the winter.
Wow, that is so exciting. This is amazing. We just really look forward to hearing the full picture from you when you come back in a month. Do you think we can expect to see a few more, or do you think it might, the period of time might be over?
The run is probably over. It is in Los Gatos Creek. It's, I should have mentioned they're also seeing this at Puta Creek, which goes through UC Davis.
Wow,
and
that's amazing.
It is quite interesting.
Wow.
Yeah.
Well, we have many of our viewers tonight walk around the lake all the time, and they've contributed the observations that I've sent to you. So I want to thank them. And everybody, just keep your eyes peeled. We may see one or two more, and we'll get Dr. Ali back in March to tell us the big picture of salmon in the Pacific, and also here what they're doing here in Lake Merritt. So thank you so much, and really look forward to your talk in a few weeks.
Thank you, and you're welcome. Be my pleasure to come back.
Fantastic. Well, as you can guess, there's a lot to tell about this story, and Dr. Ali will be back. In the chat, there's a question about will a salmon die, and which salmon swim out to the sea. Those in the Chinooks, it would be the young that would swim out from the freshwater creeks into the sea and spend some time there. And then what we're seeing is the completion of the circle of life, where those adults that grew up in the sea come in to reproduce, and they only do that once in the Chinook salmon. So huge story, and I hope you're all excited to come back and hear a lot more from Dr. Ali. So now we're going to turn to our featured main speaker for tonight, Dr. Jim Carlton, who grew up in Oakland and got his scientific start in Lake Merritt, and he's going to tell us a bit of that story and where that research is going today. Professor Carlton is a professor of marine sciences at Williams College, and a director of the Ocean and Coastal Studies program of the Williams College in Mystic Seaport. And so we're honored to have you here tonight, Jim, and are excited to hear about the lake's most famous animal, the Lake Merritt tubeworm. Happy 100th anniversary to the worm. So Jim, are you ready to begin?
Well, good evening, everyone, and good evening from the East Coast. I'll just want to tell you a short story about a worm that has a global history for now about 100 years. In 1921, a short article appeared in the Oakland Tribune simply called Coral Reefs Spreading in Lake Merritt. It begins in one short year. The city of Oakland has acquired, among other things, one handsome coral reef. And a picture here from the 1960s in Lake Merritt shows how one might think these animals were coral-like in forming these small reefs. It's hard to read this article. Here's a translation of it, as it were, which says that this hobo coral started to appear again about 1 year ago, and comments from the superintendent Lee Kerfoot, who has some long history in the city of Oakland at the time, back up in the 1940s, said it was an interesting matter for biologists. And it was. Certainly a zoologist at University of California at Berkeley soon identified it as a tubeworm, not a coral, which was not a surprise. Corals would not be expected in San Francisco Bay. A tubeworm, as many of you know, is an invertebrate. It is an annelid worm, and annelids include abundant marine worms such as pile worms, bait worms, earthworms, leeches, and now some other groups as well. A major group of annelids are the polychaete worms, which are most of the common marine worms. Not all polychaetes are marine. There are some freshwater polychaetes, and there's actually a terrestrial polychaete or two. And there are a great many families of polychaetes. One of them is called the serpulids. And these are worms that secrete and live in calcareous tubes, and they are permanent residents in those tubes. They don't leave that tube. If were they to be dislodged from that tube, they would die. Tubeworms, as with many marine invertebrates, reproduce by releasing eggs and sperm in the water. The sperm fertilize the eggs, and they develop over a matter of time into planktonic larvae. In this case, these are feeding larvae. One stage is called the trochophore, and there's a picture of a trochophore, a very small organism swimming in the water. They swim around for 3 weeks or so. They settle on a hard surface. They're not very fussy about what surface they settle on, which includes all kinds of hard surfaces. They can settle on the seaweed. They're really very broad in what they can settle on, which is part of their success. And they metamorphose into a tiny worm, which then begins to secrete that calcareous tube and cover the soft body. That tends to work out pretty well, since in some areas there are literally millions of tube worms in huge aggregations. They are plankton feeders using their ciliated tentacles, and they close that tube with a hard part called the operculum, which has some spines on it. So the tentacles go in first. The last thing in is that operculum, which may help protect them from a passing fish that would like to nibble on the tentacles. It's also a classic fouling organism. And by that, we mean it covers all kinds of things, the bottoms of boats, any submerged structure, often in huge masses, fill up drain pipes and block screens. So around the world, it has a history of being a nuisance species, and at times an attempt to control it. But back to the lake. This was new for San Francisco Bay. The bay was explored since the 1850s, and a worm producing large colonies and masses like this would have been seen before. It had not been seen before in the bay or anywhere in California. What was its name, and where did it come from? And interestingly enough, for that, we have to begin our story in Europe. In 1921, remarkably, just about the same time the Lake Merritt tubeworm showed up, a tubeworm never seen before, which sounds familiar now, appeared in Western France. Professor Mercier, a zoologist, found the tubeworms, gave them to Professor Pierre Fauvel. He's a French worm guy, specialist in the taxonomy of marine worms. Fauvel recognized it as something that he also was not familiar with, concluded it was both a new genus and a new species. This is some of the original publication in 1923. It's Says the locality where it was found was the Caen Canal to the sea, up here in northwestern France, in the English Channel. And he describes it as a new brackish water serpulid, and specifically says this interesting serpulid was collected last summer. This would be technically 1921 because the paper was written in 1922 and published in 1923 by Monsieur Mercier. So Fauvel named the new genus Mercierella in honor of its discoverer, Professor Mercier. But Fauvel thought this was a fascinating species because he himself had never seen it before, and he wondered about its history. And so he wrote some interesting comments that we find to be particularly pertinent now, a century later. What he said was that, that he lived himself on the Caen Canal in the 1890s. He had never seen anything like it in the canal, on the coast, anywhere nearby. And he speculated, which was somewhat rare for the time, that this was a species that had been introduced on the hull of a ship and that it found conditions favorable in the canal. But he said, we don't really know this until we find out where it came from. He was confident it was not European, that it would not have been missed previously, and then thought, as they were just at the end of World War I, that it would be interesting to know the history of the ships that had been visiting the Caen Canal since the war. That might provide some interesting indications. So Fauvel named it Enigmatica, since it was a mystery as to where it came from. He knew it wasn't from Europe, and thus the name Mercierella enigmatica. Several years later, Professor S.F. Light, a well-known invertebrate zoologist at Berkeley, sent specimens of the tubeworm to Professor Fauvel, who confirmed that they were, in fact, Mercierella enigmatica. That didn't solve the question, however, since European workers also said they considered it from somewhere else. So that didn't really answer it. Now we have a Latin name. In 1933, Fauvel published another paper reporting on the identification of the Lake Merritt specimens. I know some of you certainly read French. The translation is that, and he made an interesting comment, which helps us along the pathway. The translation is that they were in fact Mercierella. And a very curious comment, which we think he got from Professor Light, was that a gardener of the park thought the tubeworm had appeared shortly after the stay of a ship coming from the South Seas. Hard to kind of wrap ourselves around how the park gardener would know that and relate that, but it was an interesting comment, which Professor Light passed on as well. We're going to get back to the South Seas in a few minutes. In the meantime, specimens had come to light in the 1930s that Mercierella had in fact been collected in Uruguay in 1917, some of the earliest specimens. They weren't identified, they were simply put in a museum collection. Thus, by the 1930s, the early 1930s, the world distribution of Mercierella looked like this: San Francisco Bay, France, and it began spreading other parts of Europe at the time, and Uruguay. But it doesn't mean it was from Uruguay, because Uruguay itself had been studied for worms in the decades previous, and no Mercierella had been found there before 1917. Uruguay was not coming on the radar as a possible source of Mercierella. As the years passed, 1930s, 40s, 50s, and 60s was found in new places around the world. And again, as I mentioned, it had begun to spread now throughout much of Europe, into the Mediterranean, all the way to the Black and Caspian Seas. In the 1930s, it was recorded from Sydney and Hawaii. 1940, Argentina. 1950s appeared both in the Gulf of Mexico and South Africa, and in Japan in 1966 or so. In an unfortunate twist of nomenclature, paper was published in 1978 that changed the scientific name that we've been using for many years and was very familiar to a great many people. And the worm that had been known as Merciurella enigmatica became Ficopomatus enigmaticus. And the question is, why did that happen? Ten Hove and Württemberg concluded that Merciurella was really no different morphologically than another set of worms called Ficopomatus. And because Ficopomatus had been described by a chap named Southern in 1921, 2 years before Fauvel described Merciurella in 1923, the name Ficopomatus had what we call in taxonomy priority over Merciurella. And sadly, again, nostalgic attachment to a long familiar name doesn't really count. And so we had to let Merciurella go. One interesting conundrum about its history in California is that despite continuing to appear around the world, it would appear to be a good invader. It never left San Francisco Bay. It spread around much of the bay over the decades, found in small estuaries and creeks throughout the North and Central Bay. When lagoons were being built for housing developments in the South Bay, it colonized those lagoons, often in massive quantities. But despite being a prominent organism found on ship bottoms, despite having a planktonic larvae drifting in the water for weeks, it never appeared anywhere else on the Pacific coast until 1994. And then in 1994, it appeared and began forming masses in Elkhorn Slough in Monterey Bay. For nearly 75 years, we imagine that vessels leaving San Francisco Bay would have had the opportunity to carry Ficopomatus down to Moss Landing, Monterey Bay, and the Elkhorn Slough area. And so the question is, why now? Why, after such a long lag time in not spreading, did it appear in the slough? And the answer is, we don't know. We don't know yet. Could it be something about the slough that changed? Could it be something about the frequency of vessel traffic had changed? Very often in invasion science and invasion ecology, when invasions occur is one of the most elusive aspects of our being able to predict the movement of species. And then something else happened. After all these decades, Ficopomatus began colonizing and appearing in bays and harbors and piers and docks in Southern California in the year 2000. Work was published a few years ago, found that these new Southern California populations were more genetically diverse than those in Central California, which were more closely linked to each other. And in fact, the Southern California populations shared genes with Australian populations that were not in the Central California populations in San Francisco Bay or Monterey Bay. A reasonable guess is that at least some of these Ficopomatus are not coming down from Central California. The general trend of species is moving poleward because of climate change. We do see a few things occasionally moving equatorward, but this would be very unusual. But rather, these were new Ficopomatus coming in from overseas. The question, as with Elkhorn Slough, is why now? And so this is the picture we had by the end of the 20th century or so. A good global disperser. It in fact had colonized the Atlantic coast starting in the 1970s, 80s, and 90s, pretty much completing its global voyages around the world. One of the few missing places would be then the west coast of South America, and we would expect it in Chile, but as far as we know, it's not there. What's interesting is that in recent years, some information came to light about the even earlier history of Ficopomatus. Some of this information had in fact been around since the 1960s, if not earlier, but it had been lost in forgotten archives and letters and simply been overlooked. And the first piece of very compelling evidence was that, in fact, it had been found in Australia in the early 1900s, especially in the Swan River in Perth in 1906, and much earlier in Sydney in 1910. That alone would not tell us that the area of origin would be Australia. There are a lot of artifacts in collecting history for a lot of species that have to be accommodated. However, very interesting was that in the 1990s, and really not analyzed for some years later, subfossil tubes of Ficopomatus dated at about 700 years with carbon-14 were found to be in an estuary in southern Australia near Melbourne. There is no other serpulid calcareous worm in southern Australia in estuaries today except for Ficopomatus, and no other species in Australia of serpulid is known to have become extinct. And finally, research from a few years ago found that the greatest genetic diversity at a species level, we think, is in fact in Australia, where there are 2 or 3 distinct genetic clades, we would now call these species level entities, were centered in Australia, suggesting that the evolutionary diversity may well be rooted there. Putting all that together, it seems that very likely what we saw, going back to our original story, that during World War I, between 1914 and 1918, with a striking surge in global shipping, connecting major ports all over the world, that's when Ficopomatus left home, left Australia, showed up in South America, in Western Europe, and then in San Francisco Bay, remarkably in a very, very tight pattern. So we could call the Lake Merritt tubeworm the Australian tubeworm. That seems to be fairly certain now, where all the evidence, independent lines of evidence are pointing. And we have other species, of course, that we've gained in California from the Australasian area that we've realized over the years. Let me just wrap up by saying that it was a little over 58 years ago in 1962 that I visited Lake Merritt and happened to wander along a narrow beach right below Adams Point. The water level happened to be low that day. Had it not been low, I would have not gone down to the beach to investigate, and my personal trajectory might have been somewhat different. But as I walked along, I stepped on something that I cracked under my foot. It was a calcareous something about 4 inches wide, and I had absolutely no idea what it was. But a few weeks later, I went back to the Rotary Nature Center in the park. In a glass case on the wall was the very thing that I had found. It's now gone, it and the label, but it said Merciurella enigmatica, tubeworm from the South Seas. And I thought that was a pretty cool thing. One of the most amazing things. What was something from the South Seas doing in Lake Merritt? So I relate my visit to Lake Merritt, and especially to the Rotary Nature Center. It caused me to continue to ask questions, to Paul Cavell and Rex Burris, who encouraged me, and devote my career to study invasive species around the world for the last 50-some years. This is my original label of those specimens, and those are the specimens I collected on that September day. Thanks very much, and happy to answer questions.
I have to begin with, it's hard for me to get my mind around the idea that in 1921, these botanists or these men were in a position to identify a worm, be it in San Francisco Bay or in Europe, and say, oh, well, this has not been found anywhere else. That there was that much communication, that much level of cataloging and information that have been gathered.
Yeah.
So one question I have was, is there a marking point that we could identify in history besides the fact that 10,000 years ago, people were fascinated by insects? When that whole process began, that marks our modern era?
in terms of David, sort of documenting biodiversity and natural history, it goes back certainly for a long ways. We appreciate what the Greeks and Romans were cataloging in the Mediterranean, and we can identify species in Aristotle's works. So it goes back a long way. Modern understanding of marine biodiversity really doesn't begin until the late 1700s and early 1800s, and for an amazing part of the world, not until the 1900s. So there's lots of areas of the world where we have very little what we call baseline information. This particular species forms these massive tubes, so it was pretty recognizable. There's an awful lot of little worms, polychaete worms, that are not much bigger than a dime and much smaller, for which we could not really trace this kind of history until recently, when we now can use things like molecular genetics to try to look for what we call geographic tracks. But it goes back a fair ways. And of course, communication was at a pace that we now might appreciate. There was no email, no fax, not even phones. And so when SF Light communicated with Professor Fauvel, he wrote a letter, sent specimens to France, and he didn't expect to hear anything for a month or two. And that was the pace of life, which I do reflect on occasion.
Julie's asks, what do the Australian tubeworms eat and what eats them?
Yeah, good question. They are plankton eaters. They eat small particulate matter, phytoplankton, and doubtless some small zooplankton, and with those tentacles that reach out into the water column, and they have cilia that beat those particles down into the mouth. Who eats them? An interesting question. There is some evidence that birds do dab away at them on occasion, but it's likely that a good deal of the mortality occurs when they are in their planktonic life. And are consumed by other plankton eaters. Once they're in those tubes and forming reefs, it appears that very often they could avoid predation.
So, what effect, Martina has asked, do the tubeworms have on the rest of the ecology of the rest of the wildlife in Lake Merritt, I assume, and also the estuary?
Yeah, when Ficopomatus forms what are essentially reefs, which can be meters in extent, and they can really alter the entire community ecosystem. What we have is a calcareous worm that is really the predominant structure, three-dimensional structure on soft bottoms. They can influence the diversity of species that are there, the ones that were previously there. They also provide habitat for a lot of species, being sort of three-dimensional structures that they are. They filter a lot of water. They are excellent spatial competitors, and very often when they get to space, they hold the space and nothing else can use that primary space. In terms, that's an ecological view. In terms of economics, they are often considered around the world as quite a pest, where they can form these massive colonies that are a meter by a meter, and then interfere with whatever one is doing with pipes or grates or whatever that might be. So we don't know a lot about, and that said, about what Ficopomatus does in other situations.
So we have more questions coming. So Jim Covell was saying that he recalls them colonizing the log booms that are placed in Lake Merritt for various reasons. And sometimes they're made them so heavy that they would sink. So that would be certainly an impact on, for habitat and for, for the lake. Yes.
We don't see those large colonies as much as we did back several decades ago, Ficopomatus really flourishes in brackish water, in lower salinities.
Mm.
The lake tends to be a somewhat higher salinities very often these days. And so while Ficopomatus is still there and has been there for 100 years, we rarely see those massive colonies that used to sink the log booms around the Duck Islands. And that's a good point.
Yeah, there is a lot of change that's happened over the drought period. We've had like 2 years basically without a fresh spell in the lake, almost, and that could affect them. They seem to be not so numerous on the pilings of the docks right now than they used to be. Old photos.
Katie, I'll read the next question.
Oh, please, yes.
It's from T, and the question is, what, if any, tubeworm species are from California? And Jim, I'd like to take a stab at answering it. A short answer.
Sure.
The ones that are found in San Francisco Bay.
Yes.
Are the ones that we know of that are from California. Is that correct?
Yeah, we certainly have a fair number of native serpulids, calcareous tubeworms along the Pacific coast, from Alaska down to Mexico. And some of them are regarded as occurring in other parts of the world or very broadly in the Eastern Pacific. Few are restricted to, say, Southern California and the Panamanic region. But we do have a modest number of native tubeworms that occur along the coast, and a few in San Francisco Bay. We also have other introduced tubeworms as well, 1 or 2, I think, in Southern California in a different genus called Hydroides.
Oh, great. Thank you so much. I was rather off, but the next question from AC asked this Elkhorn Slow question. Does Ficopomatus occur in the marina, upstream of the highway, or both? In the marina, upstream of the highway, or both? And this goes to the question of the frequency of introduction into subtle habitat at the slough.
Yeah. That's an excellent question. And we have to ask some of the folks who are down in Elkhorn Slough at the National Estuarine Research Reserve if they have a good sense today as to what the full distribution of Ficopomatus is and how it's doing down there, and if it still forms some of those reefs that it did, nearly 20 years ago. I would expect it occasionally to show up, certainly in Moss Landing Harbor by export of larvae, and just the way they do in San Francisco Bay, where individual Ficopomatus show up in odd places around the bay. But I don't know the distribution of it in detail down there. It's a good question.
There's one more, Katie.
Oh, yes. We have a question from Martina. Is the salinity part of restoring the tidal flow? I think relating to the Lake Merritt Channel. Am I right, Martine? That we mentioned that the lake has become more saline than, and yeah, she says yes. So it's kind of become oceanized. Yes, go ahead.
Katie, I'll let I'll let you handle that one.
Okay, well, I think it's due to the drought periods that we've had. We've had extended periods of drought, punctuated by maybe a fresh year, but it's been very dry in California, and that's changed as Dr. Carlton said, that the Ficopomatus is a brackish-water species, so it may not do as well in the more saline waters of Lake Merritt right now, but it may come back. I had a question about where is Ficopomatus continuing to be brought in by ship, or is there an effective way to limit that and other species that might be hitching a ride by ship?
I I would assume on a general basis that Ficopomatus continues to move around on ship bottoms. And of course, every time as its global distribution increases, every time it has a new population, it interferes with, it interfaces with another hub of shipping. So with many of these species that continue to gain broader and broader global distributions, then you get into another hub of that wheel that takes you even to new routes and so on. So I would presume Ficopomatus is still a world traveler, if not on ship bottoms, also possibly in ballast water as well. And that's where the genetics comes in, where we can look at, and now retrospectively, and it'll be fascinating to do this, especially in Europe, as to how the genetics of populations around the world now relate to what are thought to be 2 or maybe 3 different clades species in Australia.
So we had a spate of really great questions here. We have a question: do the Ficopomatus clades, going to the genetics in Australia, have geographic structure that could explain the colonization of Southern California by a clade directly from Australia, rather than the clade that had been previously colonized,
Yeah.
Central California?
Yeah, my understanding is that the Southern California populations, at least those that have been sampled, do reflect at least the two distinct clades that are in Australia, and those 2 clades are not represented in the San Francisco Bay or Elkhorn Slough populations, which would give a clue that that's somewhat new colonization from Australia. But we don't think we have that global map just yet. But there's another footnote here, which is if there are 2 different species in Australia, which one is Enigmaticus?
So lots to be learned by future research. AI, I believe it is, or Al asks, can fluctuation of tubeworm colonies be considered a harbinger or an indicator of local environmental conditions, good or bad, over the past and future decades?
Fairly broad question in terms of the incredibly broad menu of environmental conditions. Ficopomatus seems to do well in shallow warm waters. They might do well in fairly productive waters, for example, and brackish water. So if some sites were becoming more brackish over time, one might expect Ficopomatus to do relatively better. Whether or not some changing salinity regimes in Elkhorn Slough might reflect that somehow, I don't know. But yes, the broad answer is certainly new populations of Ficopomatus showing up in places where they never were historically could be a reflection of shifting environmental conditions, or it could be that an opportunity was there for colonization that simply was by a vector that was not available previously. The Ficopomatus does continue to spread, which is really interesting after all these years, and there are new populations up in the Baltic Sea, for example.
That's incredible. Just, to come back to say a little bit about Lake Merritt, that there is and has been communication with San Francisco Bay over the years. Although there is a tide gate structure there, it opens and lets considerable amount of water in and out of the lake, which undoubtedly could contain these the larval Ficopomatus and lots of other things. Though I don't know about the life cycles and how long the planktonic forms or larval forms last that might bear on it. But Lake Merritt's a pretty interesting place to study, I think, from that aspect. And I know Dr. Andrew Chang at San Francisco State has correlated a lot of community structure with salinity, which we have seen some very strong swings at Lake Merritt over the past decade, really. So an interesting environmental condition. We have another question from Joan saying, does the expansion of the extent of Ficopomatus growth compared to other critters that have been recorded coming in and out, like the, for example, the clams? No, that's okay. Go ahead.
Sorry, another good question, and I'll broaden that out a little bit, Joan, which is to say that the whole picture of the variable population dynamics of the couple of hundred invaders, at least, in San Francisco Bay, really has never been thoroughly synthesized. Species have booms of populations over the years, others retract, and sort of wobble all over the place, and that certainly may be correlated with droughts and floods of San Francisco Bay, vast changing conditions of salinities, the warming of the base over the years, if that in fact is happening in the corners of the bay, which have always been quite warm. The list of environmental conditions and fluctuations in the bay potentially is very long, which could mean that hundreds of species react in many different ways. So how Ficopomatus expanding around the bay over the past 100 or so years compares to the expansion of other species, I don't have a good feeling for that. Others might, but it's a fascinating research question. I will say that we have lost species as well, not complete extinctions perhaps, but there used to be signature species in the bay that have retracted, as well as invasions that have expanded. One of those was our native horn snail, called Cerithidea, California Cerithidea, for many years. The name has changed, the genus name has changed, but evidence suggested it was probably present in vast numbers on the margins of San Francisco Bay and in Lake Merritt in the 1870s, when Lake Merritt had large tidal mudflats, which don't really exist as such anymore.
So we're going to have to take a pause here. It says, and Dr. Carlton, you're able to stay after 8 for a little more discussion?
Sure.
Okay, so we just have a wonderful, a rich amount of material for our hour, and I'd like to take this time to introduce everybody to our featured speaker for February, Dr. Frank Sulloway. He's going to introduce his research subject and as a Darwin scholar in the next small segment. So next month, we are going to be privileged to have as our featured guest, Dr. Frank Sulloway, to talk to us about the Galapagos and Charles Darwin, what Darwin would like to have known about the Galapagos. And so I'd like to introduce Frank to you. And can you tell us, Frank, what first got you interested in Darwin?
Well, when I was a sophomore at Harvard University, I read Darwin's autobiography. It's a charmingly written work. And the thing that struck me immediately is that Darwin was not a sort of a super great student. He was a relatively, I mean, he wasn't terrible, but he was sort of middle of the road. And I asked myself, gee, how is it that a sort of an intellectually average person ends up revolutionizing the biological sciences? Just intrigued me. That he struck me as a sort of a great role model for anybody who didn't have an IQ of 230 who wanted to start their own revolution.
Do you think that Darwin would have been surprised today were you to visit the Galapagos?
Well, he would be extraordinarily interested in all of the new discoveries that have come out of the Galapagos. And that whole topic actually has a sort of a fascinating relationship to the origin of species as Darwin wrote it, because there's a lot of use of the Galapagos that Darwin makes in the origin, and at the same time, there are things that you would have expected him to have put into the origin of species that aren't there. For example, there's not a single mention of Darwin's famous finches in the origin of species. It just seems like is that possible, that the most famous bird group and sort of the most famous exemplar of evolution in action brought to us by Darwin is not mentioned in The Origin of Species? And there's a very interesting story behind that, why he didn't mention the birds. On the other hand, I think it's a really interesting point if you analyze the structure of the origin, that Darwin would have been interested in things that we wouldn't have predicted. In other words, he would have wanted to add to the Origin of Species in a new edition today, things that bolstered his basic strategic argument. And those things are not the things we would normally expect him to have been interested in.
This is intriguing. I can't wait to hear next month what some of those things are. You chose to tell the story in a recent publication of yours using the Darwin's bears. Can you tell us a little bit about the bears?
Well, I've written a kind of a scientific fable, and in the fable there are 16 bears, each from a different island in the Galapagos. And they actually represent a kind of a takeoff on Darwin's finches, of which there are also 16 species. And I wrote this book originally for a godson. And when I got done with it, I thought to myself, this is much too good for a kid. I had so much fun with the story that I ended up writing what ended up as a 190-page book. And it's really an attempt to teach the basic elements of evolutionary theory, but to do it in a really fun way, and also to do it in a way that's very faithful to Darwin's own life. So the really interesting feature of the book is how you can have these bears running around in the book helping Darwin, and they all move from the Galapagos to England with Darwin when he returns on the Beagle. And yet have the story be almost exactly the same as it turned out in his real life. And that was the challenge of writing this book.
It is entertaining. I can't wait to hear more. And I've shared this with some of our listeners, a link to the book page to learn a little bit more about this. And I can't wait to learn more about what Darwin would want to know today about the Galapagos. Thanks so much. Looking forward to next month. Thank you, Frank, for giving us a preview of so many wonderful topics that we can discuss here at the Lakeside Chats. Yes, David.
Yeah, I'm sorry. On behalf of my alma mater, I got to say, go Bears. Go Bears.
Oh, yeah. Oh, yeah. Yeah, okay. So now we're coming to the end of our time, but we will have some time afterwards to after our closing at 8 o'clock, and where we can ask more questions to Jim and to the other scientists if they're here. I want to thank everyone who has made this team effort possible. Importantly, Rob Lamone, our producer, who has made videos and put everything together so that we can bring all these things to you tonight. David Wofford and I are co-chairs of Rotary Nature Center Friends, and we have a wonderful collection of members of our group who've helped make this fall possible, indicated here. We have partners, the Frederick E. Hart Foundation for Educational Opportunity, Lake Merritt Institute, St. Paul's Episcopal School, San Francisco Elks, Oakland Public Works, Youth Employment Partnership, and the schools, and the Environmental Justice Caucus of the teachers. There are programs are rebroadcast by the City of Oakland TV station, KTOP TV 10. And if you want to tune in there, you can see past shows, and the current show is usually shown at the end of the month on Sundays from 6 to 7 p.m. If you like our programs and would like to see us continue, we would really appreciate your support, and to make a donation to Rotary Nature Center Friends, you can mail a check to our post office box listed here. We will get this information to you in an email after the program as well. And we would want to express our gratitude to donors who have generously supported us so far. This is making it possible for us to do the chats and also some in-person on-site science activities with students and really volunteers of all ages at Lake Merritt. So we're looking forward to Dr. Sulliway will tell us about Darwin and his what he might have wanted to know about the Galapagos had he lived today. I want to thank our volunteers who come out and do cleanups and investigation of marine organisms, and also marsh study and restoration activities at Lake Merritt. So at the ending, we sadly would like to express our sadness that we lost an important naturalist, visionary conservationist, scientist, and writer on the 26th of December. And I included this because the E.O. Wilson Foundation actually sent us congratulations to Lake Merritt on the 150th anniversary of the Wildlife Refuge. And I want to thank all of you for coming and see you not in January, but in February. That slide didn't get updated. Okay, so I know. Well, so that brings us to the end of the slides, and maybe we'll take about a 1-minute break, and then please stay with us, and we'll continue. We'll check in and see who's here and continue with our questions. Okay, thank you so much. Let's take like 1 minute. So what we normally do on Lakeside Chats is we have questions and answers and discussion. We can talk about any part of the program, and you can suggest topics that you think would be good for us to pursue in the coming weeks. I see we have Dr. Ali is with us, and of course we have Jim Carlton, and I'm not sure Frank's here, but anyway, we could ask questions. And so please either raise your hand or put a question in the chat. We'll go on until 8:15 or.
Raise my hand, Katie.
Pardon?
I want to raise my hand.
Yes, David.
Actually, I'd like to pick right up where we left off.
Great.
Dr. Carlton responding to the last question, which he began, that it was a broad question, and I felt as if it was so broad, it began to answer the question that I would have been proposing or sharing, and that is that earlier I began to ask leading questions about the history and the background, and the fact that human beings have been studying and fascinated and collecting data thousands of years, and that there eventually was moments in time when things became much more active and serious and engaging, and leading up to my idea that the period from what I'm hearing, between 1850, let's say, and 1930 around the world, there was this, I wanted to know if it would be fair to say, a huge explosion of activity and human beings that were involved in this dynamic of gathering information. And I wanted to, I guess, pause to know that that was true, but then go on to say, there you have 1921 and the tubeworm in Lake Merritt, not seen before, all this cataloging and body of information, and then to France, and then eventually the other identifications and locations, all the way down to Southern California today. And yet the question about, so for me, when I heard it described, there was a lot of the core of my question was, wow, how do we even arrive at a point where after all of that, something could happen in these modern times where there could be something living in Lake Merritt or San Francisco Bay for, what was it, 80 years. And then all of a sudden it begins to move. And where does that leave us? And lastly, and I'll stop here for Dr. Carlton's comments, in his response to the last question, is it simply because we haven't fairly synthesized the experience out there that we're looking at and talking about? Is that the core of that? Dr. Carlton?
Well, yeah, David, you've touched on a lot of interesting questions about the history of exploration, about how our knowledge expands, whether it's in any environment, in the ocean, on land, or in freshwater systems, and the participation of the public as well. And I'm not an expert in a number of the fields in which you touched upon. Of course, that rarely prevents us from commenting on it.
Well, keep it simply to why would something move that hasn't moved?
Yeah.
And
I'll do that. I'll tackle that one. But I will also say that there's an awful lot that is in motion, that is moving, that we simply don't see, we don't detect. And there are cases across my desk almost every day of reasonably large species that are within arm's reach, elbow reach, that are all around us that no one has even commented on. So, and that's in the coastal environment. So I can talk about that a little bit later. Why do species continue to move? Overall, one of the grand scale effects right now that we see is that there are a tremendous number of species moving poleward in both hemispheres because of climate change. So there are a lot of species moving north along the, for example, where I am on the Atlantic seaboard, we call it Chesapeake creep up here in New England. These are species that have classically been no further north than Chesapeake Bay, that are moving into Long Island Sound and the Cape Cod region, Gulf of Maine, rather steady stream, often some eyebrow raising species that we thought simply could not survive in what was a formerly cold New England. Down south, we have Caribbean creep, which are lots of species moving up from the Caribbean onto the south US coast. So climate change is a reason why species are moving in many cases. Habitat change can also affect why species move. And then in general, much more broad stroke, is that we still have tens of thousands of ships moving around the world on any given day. And they are carrying an unknown number of species, either on the hulls or in the boats. And opportunity presents for new colonization all the time. And some of those colonization events are simply the right combination of the amount of propagules, the larvae, whatever it might be, that are arriving in a particular environment at the right moment. And you might land in a place in the winter when summer would have been a better time to colonize and that sort of thing. So there are a lot of seemingly random events, but the bottom line is we continue to get new invasions around the world. I can speak to the marine environment of species that for all intents and purposes seem we should have shown up decades ago. And we just don't know why they now come in. All of that leads us to one of our fundamental strategies, which is knowing that species continue to invade, species that we thought should have invaded decades or even longer ago continue to invade. Opportunity always presents, will continue to present, which is why preventing invasions tends to be one of our most often pressing strategies.
Thank you.
Fairly broad answers, actually. Sorry.
Well, you spend a lot of time actually with the with some of the setup, because to some extent we start with the understanding that species movement around the planet and among throughout species can result from food availability, temperature change, climate change, vessel movement, and predator. And all of these things have been in effect in San Francisco Bay and the worm has been in the bay. And now we're aware that it's gone. I mean, that is, I'm sorry, moving around. And
Yeah.
then as you pose, so, oh, I forgot to add one other thing that causes movement. That is landlords raising the rents. uh, um, and ultimately, I couldn't help but consider, don't mean to get too off track, how, you know, when we look at economies and our system and see how it has to do that and how we seem to be trapped in it, maybe our focus needs to be more on prevention, like in the environmental world when it comes to this kind of study as well.
Yep.
But nevertheless, I was just amazed that after Darwin and all of the collection of data, that we could find ourselves in a moment where something could be happening and is happening there that we don't know about right there in the bay.
You know, we continue to get new invasions, as we've been talking about, all over the world. Here on the Atlantic coast, we have no fewer than 3 new species of shrimp that have appeared in the last 20 years that are doing quite well. One is from Japan, 2 are from Japan, and 1 is from Europe. And the European one, which we first detected as recently as 2010, is now the most common tide pool shrimp nearby me on a rocky shore. And it was simply not here. It's a native European species. I suspect it's been coming over for an awful long time in ballast water, but we first found it in 2010. So I expect to find new invaders here in New England once every couple of years at least. And predicting what those will be and where they might be from continues to be somewhat elusive. We do have a list of species that we expect to arrive. They're on what we call watch lists, and we put out color cards and brochures and put them up on websites. But often as not, the species that show up are not on our watch lists.
I've been on a few watch lists myself, but I want to invite our listeners and the people that are still with us to please join in with any thoughts and questions that you may have.
We have a great question from Naomi. She wants to know what kinds of prevention are you thinking of that might be effective in preventing invasions or movements?
Yeah, good question, Naomi. Our first matter on that agenda is to try to understand what vectors are bringing species into a region of interest. If we're interested in San Francisco Bay or California in general, or if we work in Chile or the Hawaiian Islands, wherever it might be, asking what brings species from other areas that are not yet present. And then we try to identify what those vectors are. In a number of areas of the world, there's surprising lack of knowledge of how species are arriving, and even if we know those vectors, what species are arriving with those vectors. In areas that have large ports and harbors, we expect that species are coming in both in the ships, in ballast water, which may be variously treated or exchanged on the high seas, or not, perhaps, under any number of conditions. The species that are attached to the hull of the vessels, and which has a lot of opportunity on hulls for all kinds of special niche areas to successfully come across. We're looking at live seafood imports and what you might be able to buy alive in seafood markets in large cities, Los Angeles, San Francisco, and walk out of the store with your live mussel or snail or whatever it might be from someplace else in the world. And then you could choose to just simply dispose of it in San Francisco Bay or Los Angeles Harbor. So the list of vectors tends to be surprisingly long. And then the question is, how do we manage those vectors, and what might be the economic impacts of managing those vectors? And a good example is the aquarium industry, which is a huge multi-billion-dollar industry which moves live invertebrates and live fish and live plants all over the world. And we expect a number of invasions have occurred because those species have been intentionally released in the environment by people who no longer want them as pets, or escape in some other way. So the movement of commercial oysters used to be huge. I think it still happens on some level, and we're interested in and other seafood, other live seafood for aquaculture and mariculture purposes. Not only the species that are moved, but also what's with those species as hitchhikers. So we can go on for an hour's lecture about vectors, but many of them do focus on what's the vector, what species are being moved, and can we prevent the accidental movement of species that would possibly cause economic, ecological, or public health concern.
Couple of more questions, Katie. You got them? I got. Here's one.
There's one. Someone asked, where can you go look for those watch lists that for your local area or so forth?
Yeah, they may appear. A good question. They may appear under different names. Well, I would just Google invasive species watch lists. I imagine those are different for terrestrial plants, for insects, freshwater aquatic plants, that kind of thing. Many agencies have those watch lists up on their website at a county, state, sometimes a federal level, for looking for species of concern that are spreading or are here and are spreading or are not yet here. So try Googling watch list and with the word invasive species and see what you get. There's something also called horizon scanning, which is a phrase used more and more to get an idea of what future invaders there might be.
Cautionary note, don't be surprised if you find your own species on the list.
Yes.
So we do have a question about the incidence of invasion along coast versus in the open ocean. You have any thoughts about that? I know you've done some work on rafting of tsunami material, etc. It seems interesting question.
Yeah, that's a good question, and it's a question that comes up all the time among invasion workers as to the relative distribution of invasions, where they are in which habitats, estuaries, bays, ports, harbors versus open rocky coasts versus subhabitats like kelp beds or eelgrass meadows or mangroves or coral reefs. We find invasions essentially in all of those habitats, including the deep sea, but relative numbers vary hugely. The open ocean question is an interesting one. For those of you who are still with us, if you'd like to Google the word neopelagic, Neopelagic, neopelagic. We published a paper in early December suggesting that, in fact, the open ocean may be increasingly colonized, and by open ocean, I mean the high seas, the mid-ocean, by coastal species that have found plastic as a suitable habitat out there. And the vast amount of plastic floating in the ocean, virtually semi-permanent, we have evidence that coastal species are surviving out there and reproducing out there. We used to think that the limitation was physiological and that they couldn't survive out there, but in fact, maybe one of the primary limitations was there wasn't much place to live out there. So neopelagic, we've just published some work arguing that the open ocean, the high seas, may in fact be subject to invasions. Much harder to study, of course. We have access to the coastal zone, and the estuary is not easy to get out there in the mid-ocean.
So Jim, are these materials in the open ocean? Are they how would they be colonized from coastal areas? Are there planktonic forms or whatever that get there, or they have to run into another piece of wood or that has something?
Yeah, we can use the tsunami incident of March 11, 2011 as an example where millions of items got ejected into the North Pacific Ocean, and a fair number of those got colonized or were colonized by Asian species, and a lot of that material drifted over to the Hawaiian Islands and to North America. and it's in that transit, those species spend a lot of time, and we begin to realize years, in the North Pacific gyre and in other parts of the North Pacific. So essentially they have to exit the coast in one way or another, and then find themselves out there, and then reproduce or and get entangled, so that a species, that an object from Asia with a living Asian species would get entangled with fishing nets, and then those species would simply colonize those fishing nets by either direct transfer or by reproducing and settling on yet another object that did not come originally from the coastal zone. Yeah, it's a whole new field of study where we're just beginning to look at this. It's quite amazing.
We had a question specifically about a species called Bidulphia, or Biddulphia. Would you consider that to be an open ocean invader?
Yeah, interesting. I'm a little rusty on the distributional ecology of that particular diatom. We do have an open ocean invading diatom, a surf zone diatom in the Pacific Northwest that I suspect came from Australasia back in the 1940s and 50s. But I don't recall whether Bidulphia is in the open ocean or not. Certainly a lot of work on it. So sorry, I can't really recall the details on that one.
Let's see. Scanning the chat. Oh,
there's one here. There's one here that says, "Sorry if this has been asked already. I stepped away. Where would one get started if one wanted to work in invasion work? What does the field look like? That is, what jobs are available to people studying these things?"
Yeah, I'll frame that in terms of habitat again. There's a lot of work on terrestrial non-native species, a lot of concern about invading plants and insects. And in freshwater, a number of bivalves have invaded. What are called aquatic plants have been big invaders around the country and around the world, and a lot of species in the marine environment. These tend to be divided up into whether one's working in marine, aquatic / freshwater, or terrestrial environments. That said, there are strong academic approaches where one does research as an undergraduate or graduate student, or as a professional researcher looking at a particular species, its ecology, its biology, its distribution, its physiology, its genetics, and or including looking at management issues, management and policy, how to control the spread of species and or prevent their invasion. So it's a pretty broad menu. Along that line, then, what jobs are available depends upon whether one wants to go into academic research, go into some aspects of management and policy, and so on. So to get into invasion work usually begins on some kind of an academic track, but not always, of course. And there are a lot of county and state agencies, federal agencies, that have a lot of focus on invasion management, and that varies a little bit by presidential administration. But now we have an administration that is doing more work on invasions than the previous administration. So opportunity may be there, and I do see job ads cross my desk on a weekly basis for people, for those interested in working on invasions of all kinds, including monitoring invasions and some management work. There's a great place that those are listed. It's called the Ecology List Serve, and there are regular postings, it seems to me, almost every day for looking for student research assistants and graduate students working on invasion questions.
Great.
we may have tired people out at this hour. It's
I
only
think it's, yeah, this has just been a wealth of information, and I'm sure we'll have questions coming to mind long after, especially when we go down to Lake Merritt and see all of the marine life and other life down there interacting and so forth.
Yeah.
What extinctions, have there been any extinctions in Lake Merritt?
Well, Ceratidia, the California horn snail, used to be very abundant there more than about 150 years ago, and it's not there at all now. So, but the lake has remarkably changed, of course, since its version in the 1870s, when it had these huge mud flats and probably a lot of sewage going in as well. So I would expect there was a fairly striking shift in what used to be there. Wait, I do see a question about the ecology list server. Stand by. I will. It is an active server, so I can give you some kind of a link here. Let's see here.
Great.
It's called. Let's see. Here, I'll put it in the try something like that.
Ah, nice.
Okay.
Okay, well, do we have any more questions out there? We have a few more minutes. It's really late on the East Coast. And,
the night is young. It's only 11:26.
Okay.
Out here.
We do have so we have there's Dr. Ali is here as well. If you have a salmon question that's been waiting all hour, we could ask him. Dr. Ali, I'm just going to ask one because I think it's something that's interesting. Are Chinook salmon the only salmon that that end their lives after one reproduction, or simultaris, or is that something that's more general in salmon? Oh, you've got to unmute yourself. Okay. Oh, he's talking. Brian, you need to unmute yourself. Okay. Let me ask him to unmute with nothing. Okay, well, maybe that one can wait for the next show. And
I got a belly full of salmon questions.
Yeah.
Yeah.
Yeah. Okay, Brian. You need to unmute yourself? Let me see if I can ask you one more time to do that. It's can be tricky.
We're coming up on 8:27 now as well.
Yes. Okay. I think maybe what we should do is postpone that, and it's a rich topic till March, when we can so we can keep our questions going for that. And Jim, thank you so much for a wonderful program. There's so much to think about, and
quite welcome.
Yeah,
I've got
really
something I want to say in closing to Jim. Well,
David.
Well, Jim, earlier in your talk, you mentioned visiting Lake Merritt and making a discovery. And I won't bother to re-mention the year you did that. But anyway, I was born that year. And I choose to believe that if you hadn't have took that walk and made that discovery, my entire life would have took a totally different trajectory.
Thank you, David.
Thank you so much for joining us.
That's great. Okay, well, I guess we've come to the end of our Lakeside chat, and thank you so much, everyone, for coming. Thank you to our wonderful guests, and we're looking forward to next month. And again, thank you so much. So we'll send you a recording probably in about a week.